Good morning, viewer. Welcome to Officer TV. Here's a breaking news. Yes. In Boston labs, all blind mice have regained their eyesight, developed smarter, younger brains, and built healthier muscle and kidney tissue. On the flip side, young mice have prematurely aged with devastating result to nearly every tissue in their bodies. The experiments show aging is a reversible process capable of being driven forwards and backwards at will, said anti-aging expert David Sinclair, a professor of genetics in the Blavatnik Institute at Harvard Medical School and co-director of the Paul F. Glenn Center for Biology of Aging Research. Our bodies hold a backup copy of our youth and can be triggered to regenerate, said Sinclair, the senior author of a new paper showcasing the work of his lab and international scientists. The combined experiments published for the first time Thursday in the journal Cell challenge the scientific belief aging is the result of genetic mutations that undermine our DNA, creating a junkyard of damaged cellular tissue that can lead to deterioration, disease, and death. It is not junk, it is not damage that causes us to get old, said Sinclair, who described the work last year at Life Itself, a health and wellness event presented in partnership with the CNN. We believe it is a loss of information, a loss in the cell's ability to read its original DNA, so it forgets how to function. In much the same way an old computer may develop corrupted software, I call it the information theory of aging. Zha Huan-Yang, a genetics research fellow in the Sinclair lab who co-authored the paper, said he expects the findings will transform the way we view the processes of aging and the way we approach the treatment of diseases associated with aging. The epigenome is the software. Epigenes are proteins and chemicals that sit like freckles on each gene, waiting to tell the gene what to do it, and when to do it, according to the National Human Genome Research Institute. The epigenome literally turns genes on and off. That process can be triggered by pollution, environmental toxins, and human behavior such as smoking, eating, an inflammatory diet, or suffering a chronic lack of sleep. And just like a computer, the cellular processes becomes corrupted as more DNA is broken or damaged, Sinclair said. The cell panics and proteins that normally would control the genes get distracted by having to go and repair the DNA, he explained. Then they don't all find their way back to where they started. So over time, it's like a ping pong match where the balls end up all over the floor. In other words, the cellular pieces lost their way home, much like a person with Alzheimer's. The astonishing finding is that there is a backup copy of the software in the body that you can reset, Sinclair said. We are showing why that software gets corrupted and how we can reboot the systems by tapping into a reset switch that restored the cell's ability to read the genome correctly again, as if it was young. It does not matter if the body is 50 or 75, healthy or racked with disease, Sinclair said. Once that process has been triggered, the body will then remember how to regenerate and will be young again. Even if you are already old and have an illness. Now, what the software is, we don't know yet. At this point, we just know that we can flip the switch. The hunt for the switch began when Sinclair was a graduate student, part of a team at the Massachusetts Institute of Technology that discovered the existence of genes to control aging in yeast. That genes exist in all creatures, so there should be a way to do the same in people, he summarized. To test the theory, he began trying to fast forward aging in mice without causing mutation or cancer. We started making that mouse when I was 39 years old. I am now 53, and we have been studying that mouse ever since. He said, if the theory of information aging was wrong, then we would get either a dead mouse, a normal mouse, an aging mouse or a mouse that had cancer, we got aging. With the help of other scientists, Sinclair and his Harvard team have been able to age tissues in the brain, eyes, muscle, skin and kidney of mice. To do this, Sinclair's team developed ICEs short for inducible changes to the epigenome instead of altering the coding sections of the mice DNA that can trigger mutation. ICE alters the way DNA is folded. The temporary first healing cuts made by ICE mimic the daily damage from chemicals, sunlight and the light that contribute to aging. ICE and ICE mice at 1 year looked and acted twice their age. Now it was time to reverse the process. Sinclair lab genetist Yang Cheng Lu created a mixture of 3 or 4 Yamanka factor human adult skin cells that have been reprogrammed to behave like embryonic or pluripotent stem cells capable of developing into any cells in the body. The cocktail was injected into damaged retinal ganglion cells in the back of the eyes of blind mice and switched on by feeding mice antibiotics. The antibiotics is just a tool. It could be any chemical really. Just a way to be sure the 3 genes are switched on, Sinclair told to the reporter. Normally they are only on in very young developing embryos and then turn off as we age. The mice regained most of their eyesight. Next, the team tackled brain, muscle and kidney cells and restored those to much younger levels according to the study. One of the breakthroughs was to realize that if you use the particular set of 3 pluripotent stem cells, the mice don't go back to age 0, which would cause cancer or worse, Sinclair said. Instead, the cells go back to between 50% and 75% of the original age and they stop and don't get any younger, which is lucky. How the cells know to do that we don't yet understand. Today, Sinclair's team is trying to find a way to deliver the genetic switch evenly to each cells, thus rejuvenating the entire mouse at once. Delivery is a technical hurdle, but other groups seem to have done well, Sinclair said, pointing to 2 unpublished studies that appear to have overcome the problem. One used the same system we developed to treat very old mice, the equivalent of an 80-year-old human, and they still got the mice to live longer, which is remarkable. So they have kind of beaten us to the punch in that experiment, he said. But that says to me the rejuvenation is not just affecting a few organs, it is able to rejuvenate the whole mouse because they are living here longer, he added. The results are a gift and confirmation of what our paper is saying. What is next? Billions of dollars are being poured into anti-aging, funding all sorts of methods to turn back to clock. In his lab, Sinclair said his team has reset the cells in mice multiple times, showing that aging can be reversed more than once, and he is currently testing the genetic research in primates, but decades could pass before any anti-aging clinical trials in human beings get analyzed and in safe and successful scale to the mass needed to the federal approval.